Good morning, everyone. I am Ashpit Kaur, and my partner name is Poojan Karki, and here I am going to present our project, which was determining the efficacy of three commercial wipes against Asorius and E. coli. Firstly, I am going to start with the introduction. Surface can easily be contaminated by hands, aerosols, or fluids containing bacteria, fungi, and viruses. Pathogens can persist on inanimate surfaces for a certain time interval. Serves as important carriers for transmission of pathogens in the hospital or any other settings. It also increases morbidity, mortality, and economic loss. And here are the methods of disinfection of the surface. Disinfection can be done by spray and wiping, dipping and wiping, and soaking and wiping. It also possesses some risk factors. Formation of droplets of disinfectant in the air, which can be inhaled by workers working there and remain on surfaces. Ready to use disinfecting wipes, pre-saturated towelette, and pre-wrapped disinfectant by a wipe commonly used for the disinfection of surface and device in food, hospital, and domestic environment.
The main advantages of the commercial wipes are they are cheap, no preparation time required, safe for disposal, and have been proved by much research to have good microbial effect in different conditions.
Some factors affecting the efficacy of disinfectant wipes. First one is organic load. Second one, target microorganism. Third one, application method. And the last, type of disinfectants.
There were three main objectives of the experiment. First one was to measure the efficacy of the three different commercial wipes to remove the bacterial contamination from the surface with the help of the mechanical force and determine the kill percentage. Second one, to measure the bacterial transfer from the wipe to assess the transfer risk associated with disinfectant wipes. And third one, to measure the bactericidal activity of the test disinfectant wipe by direct inoculation and to determine the kill percentage. The main hypothesis of the experiment were the kill percentage of disinfecting wipe will match the label claim of the manufacturer. Second, the mechanical force employed by using the disinfectant wipes aid in the removal of the microbes from the surface, which results in the greater reduction of the bacterial load and hence greater efficacy of the disinfectant wipes. Third, the disinfectant wipe can prevent the transfer of the pathogens to clean areas. And fourth, the actual bactericidal activity of the commercial disinfectant wipe to kill the microbe will be reduced in the absence of the mechanical force. Now, moving on to the materials and the method, two different bacteria were used. One is the Staphylococcus aureus, which is gram positive, and next one is the E. coli, which is gram negative. They both are normal flora of the human body and acts as an opportunistic pathogen. The disinfectant wipes that were used were Lysol wipe that contains citric acid, Clorox wipe that contains dimethyl ammonium chloride, and the alcohol wipe that contains alcohol as an active ingredient. These are the list of the media and the materials used in the experiment. TSA was used for the plate count, lithium broth with twin AD was used for neutralizing, phosphate buffer for cereal dilutions, and sterile water to moisturize the control wipe. Aluminum foil was used as a carrier and the cotton cloth was used as a control in the experiment. Bacterial inoculum was prepared by mixing the pure culture with the PFO buffer. First, initial assumption of 1.5 into 10 raised to the power 9 CFU per ml was made with McFarland standard, and the actual concentration was determined by the split plate method. For measuring the efficacy of the test disinfectant with physical removal, Two aluminum foil were inoculated with 20 microliter of the bacterial sample. One is the test and one is the control. They both were dried at 37 degrees centigrade for 25 minutes. After drying, the disinfectant wipe is applied on the test aluminum foil and the control wipe is applied on the control aluminum foil. After wiping for the 10 seconds, the aluminum foil are transferred into a tube containing 10 ml of the neutralizing broth. Then the serial dilution is performed for both the sample and the control wipe, and the plate count is done by the spread plate method and incubated at 37 degrees centigrade for 24 hours. After the incubation, the plates are counted for the number of the colonies on the plate, and the efficacy was determined by the reduction of the bacterial load from the initial concentration, and also by determining the kill percentage. Measure the transfer risks from the wipe. After the application, the wipe are transferred to a nutrient broth containing neutralizing broth, and they are incubated at 35 degrees centigrade for 24 hours, followed by spread plating and then incubation at 35 degrees centigrade for 24 hours. Finally, the colonies are counted to determine the transfer risk. To determine the bactericidal activity, one test and one control wipe is inoculated with 20 microliter of the bacterial sample. They are allowed for 10-second exposure, followed by transferring to neutralizing broth. After 5 minute neutralization, contact serial dilution is performed and the spread plating is done. The plates are incubated at 35 degrees centigrade for 24 hours, and the bactericidal activity is determined by comparing with the control and determining the kill percentage. This is the result for the actual concentration of the S. aureus and E. coli in the sample. The result shows that the concentration of the E. coli was 2.1 into 10 raised to the power 8 CFE per ml, and the concentration of S. aureus was 3.0 into 10 raised to the power 8 CFE per ml. The given table shows with the mechanical removal, the E. coli concentration was decreased from initial concentration of 2.1 into 10 raised to the power 8 CFE per ml to 1.0 into 10 raised to the power 1 CFE per ml after the application of the Lysol Chlorex and the alcohol wipe separately. And whereas the S. aureus showed no growth after the application of the Chlorex wipe, and the bacterial load was decreased from 3.0 into 10 raised to the power 8 CFE per ml to 1.0 into 10 raised to the power 1 CFE per ml in case of the Lysol wipe, and 3.0 into 10 raised to the power 1 CFE per ml in case of the alcohol wipe. Kill percentage of E. coli and S. aureus was determined after the application of the Lysol, Chlorex and alcohol wipe, and the kill percentage for both of the bacteria by all the three wipes was found to be 99.99%. These photographs were taken during the experiment, and they show the efficacy of alcohol wipe and the Chlorex wipe against E. coli and S. aureus respectively. These are the plate count results obtained after the direct inoculation of the S. aureus in three different commercial wipes. The kill percentage of E. coli and S. aureus by Lysol, Chlorex and alcohol wipe was found to be 99.99% with the direct inoculation. This picture shows the bactericidal activity of the Lysol and the alcohol wipe against the E. coli. Hence, all the three wipes were found to be effective against S. aureus and E. coli with 99.99% kill effect. Since the kill effect for all the three wipe is 99.99%, so there is no any significant difference between the bactericidal activity of this wipe and the removal of the bacteria from the contaminated surface not really depend on the physical force that we apply, but the actual removal is due to the bactericidal activity of the wipe. Furthermore, a few colonies were recovered from the wipe, hence the wipe poses the transfer risk factor. So if the same wipe is used between the contaminated and the clean surface, there might be the chances of transfer of the bacteria from the contaminated surface to the clean surfaces. Finally, these are the list of the references that were used to make the PowerPoint presentation. Thank you everyone for listening our presentation and being a part of it. Thank you so much.